So this is an explanation of figure 5.12. And what we have here is a diagram showing the stable isotope ratios that are associated with plant material from different sources. So for example, we have terrestrial plants in the yellow box towards the left hand side of the diagram. The stable isotope ratios associated with freshwater marshes in the green box, then the stable isotope ratios of salt marshes, and also benthic microalgae. And this is shown for two sections of the estuarine system, the upper estuary and the lower estuary, which is shown in the lower panel. And what we can determine is the source of food that is utilised by either pelagic organisms, so they're the organisms shown by the open circles, or benthic organisms, which are the organisms shown in the closed red squares. And what we can see in the upper estuary, these two different types of organisms, pelagic and benthic organisms, in the upper estuary are deriving most of the detritus on which they feed from the terrestrial system or fresh water marshes, for example. When we move down to the lower section of the estuary, we can see that pelagic and benthic organisms are deriving most of their detrital material from benthic microalgae and also salt marshes, which understandably are more prevalent towards the lower segment of the estuary. So stable isotopes are a very useful forensic technique whereby we can look at the composition of animal tissues and determine upon what they have been feeding and what the origin of that material might be.